There's been extensive review of these chemicals by the FDA, OSHA, and the NIH. HGI contacted them, and they provided official statements. The FDA does not regulate these devices as a category because they are imitating a natural process. And so they have chosen to review all of the devices individually, and they've come back with that statement. We did get a statement from the NIH that they researched at our request all of the databases, including OSHA, NIH, FDA, and all their NIOSH and other resources, and they've come back and they said there is no evidence or data to support the fact that hydroxyls are anything but safe for plants, animals, and humans, and they see no difference between natural hydroxyls and those generated by UV optics because the process is identical. HGI was concerned that there was a lack of research that was available in the literature, and so they chose to do their own toxicology studies. And what they conducted was a 13-week GLP, which stands for Good Laboratory Practices, FDA study involving a statistically significant number of rats in which they exposed them to extremely high levels of hydroxyls, twice what we would normally encounter indoors, continuously for 13 weeks, day and night. There were, of course, controls for the study. And after that intensive exposure, the report was that there was no difference between the control population and the treated population, except for the observation that the treated animals seemed happier. And that probably was because the hydroxyls were able to kill off the ammonia produced by their waste products. And so actually it made for a better husbandry environment for the test animals. So typically when there are negative results for a 13-week study of this type, the FDA would not ask for any further studies. And so that was a very positive result. It was done by a third party called Comparative Bioscience that is licensed to conduct these studies, so that it was entirely independent of HGI. Typically with these studies, the test animals are exposed 24/7, which was the case in this study. So the device ran continuously, and it was remotely monitored to be sure that we knew what the oxidant levels were and that the device was functioning properly for the entire 13-week period. Their laboratory technicians would come and inspect the animals periodically through the day and through the night, and regular notes and observations were made throughout the 13 weeks. The animals were sacrificed, and they were studied histologically and at a cellular level as well, so it wasn't just gross observations or growth pathology. And there were no histological changes, no cellular changes at all, which is very important because you're concerned about the effect of breathing them in. And the lung tissue and the tissue in the nasal passages and mucosa were perfectly normal after this exposure. Well, in addition to the toxicology studies, HGI has conducted extensive chemical and microbiological studies. So it's important for the FDA, when they look at these results, to assess your statements. Is it both efficacious, is it effective, and is it safe? So the toxicology study addressed the safety issues, but concurrent chemical studies that were conducted independently with a major research institute called the Lovelace Respiratory Research Institute were able to confirm that the effects of the hydroxyls chemically were very effective. And so they studied the decomposition of controlled amounts of volatile organics that are well documented in the literature. And after a careful study of the kinetics in the test with the literature data, we could conclude unambiguously that it was the hydroxyls that were the reactive species and that they were being produced at the kinds of concentrations and rates that were commensurate with those found in nature. So we had both the chemistry side, we can make it, it's effective, and the toxicology side, it's harmless to people, animals, and plants. It all is a function of scaling the optics. If you think of these optics as having the capacity to generate a certain concentration, if I have 2 of them, or if I have 20 or 40, then I can scale up from a single room device to a large unit with as many as 48 of these very powerful optics that can handle very large commercial spaces, 30, 40, 50,000 square feet of space that could be anywhere from 8 to 20 feet tall. It's a question of just having the fan capacity and the optics. So there are units from every part of that spectrum.